trying to get an idea of currents a little bit. Can you tell by flying the ROV? Are there strong currents here?
They're having a quick chat.
Sorry, what was that, Amanda?
Okay.
Currents?
I was wondering if you knew anything about the currents based on how it's flying.
It's flying pretty all right. Don't really feel too much in our faces right now.
Can we get a zoom on that coral on the left that we're just past?
Yeah. We can do a really quick one, and then we're gonna have to get up this hill.
No worries.
Otherwise, I'll fall behind.
Yeah. Okay.
All right, Marley, go ahead.
Yeah, and just to remind everyone, this is definitely a dive we're trying to kind of run quickly. We're not trying to stop too much. But
Full wide, Marley.
Just as good on those snap zooms.
So Meredith in the science chat writes that this could be Calyptrophora or Narella. And then there was a chrysogorgid on top. Erica pointed out upper part of the screen.
very cool. Thank you.
All right. We'll come up to get in front of you a bit.
So there's another, kind of look more like the chrysogorgid to the left. Now there's an Antipathes, another black coral. Ooh, another one of those brilliant purple sea cucumbers. And Bathypathes to the far left, another black coral.
Yeah, so once we get up to this depth contour for A2, we'll just keep to the east side of that local max here. That sounds good.
Roger. Sounds good.
Thanks. So are any of those really deep purple sea cucumbers, the holothurians, are they any of them the pelagic species? No, right? The one that,
I don't know the answer to that. Unclear.
oh, yeah, I should have mentioned it when
We'll
it was in
see
the
if
camp.
anyone, yeah. We'll see if anyone in the science chat says differently, but
It's a little frilly on the bottom. definitely flattening.
No.
It's a beautiful stalked crinoid feather star. So all of those projections sticking up are arms of the crinoid, with smaller projections sticking out from those called pinnules. Each of those pinnules have tiny hair-like structures called cilia that beat and help direct particles that get captured or caught, and moves them down toward the mouth that's at the center of all of those arms.
So it's kind of a long chain gang there, working?
Yeah, exactly. It's like the bucket brigade, moving food further and further down toward the mouth. Oh, there's a little stalked sponge over there. Sponge on a stalk. Those are always cool. Actually, some research that I've done recently with Ken Smith's group and the long time series that's been monitored at Station M at Mbari finds that those stalk sponges will contract and then expand back up to full size. So they may look static. The only problem is that you need to be there long enough to see them in action, and that contraction takes about 4 days to happen. And then they stay contracted for about, I think it was 11 days, and then they can expand back up. So it's a slow process. They live on a different time scale than we do. But that's what time-lapse was great for.
Amazing.
You know, long-term study site, we had that view. Oh, sitting majestically on that rock to the right is a squat lobster. These are mobile crustaceans, not quite lobsters. Some people think they look like crabs, but the tail is actually curved underneath the body. Oh yeah, that's a nice view of that stalked crinoid. You can really see the arms with all the pinnules sticking out.
Yeah, so we're matching up to the map right now. Looks good.
Let me know if I'm nerding out too much about invertebrates. It's a hobby.
No. Not at all. And besides, most of what we see down here are invertebrates anyway. I mean, obviously a few fishes and crustaceans, but...
It is true. Aha, that's that coral again. Hopping back. I think, we'll wait until they write, but I think this looks also like that Calyptrophora or Narella that Meredith had identified before.
Riley, you want to snap zoom there, please?
Yeah, definitely.
That's great.
Oh, that is beautiful. Meredith says she's checking through her references to see if it is Calyptrophora.
All right, Riley, full wide, please.
Thank you.
Thank you.
Yep, sure thing.
Here we go, here we go.
We have some ideas going in the science chat to figure out who that is. So we'll let you know if we get any confirmation. Calyptrophora, between Calyptrophora or Narella. There's another squat lobster, also called Galatheid.
So I'm going to bring us to 030.
Seems like that'll still go a little down, so it doesn't look...
It may, I just, yeah, we can wait another 30 meters. I just don't want to have to maneuver around this ridge once we're on it.
And it's the first mushroom coral we've seen.
Yeah, because it seems like it'll still rise a bit in front of us. And
Oh,
if it goes
yeah.
030, it'll probably take us a little off. Maybe let's try waiting out one more move and then seeing how we flat. Great, thanks, Ben. A 010.
Or, sorry.
Thanks, Ben.
As we're surveying along here, we're passing by more of those tube worms there on the right, the ones with the flared tubes. Interesting, they seem to be on the undersides of rocks. Maybe there's too much sediment on the top.
Is that some old dead sponge material, you think, Amanda, that's been collecting on some of these rocks? They don't look like sediment. It looks like some remnant.
Yeah. It does look like
that was
remnants,
blowing.
but
That
a
one
little...
was...
Yeah, I think it's more floppy. There are a lot of what I think are still unhealthy looking sponges in this area. I wonder if the currents changed here so that the sponges that used to live here, like that one on that rock covered in crinoids, maybe used to be healthier. Unless this is a species that's just that buff color, but it's just not a common color that I'm used to seeing in a healthy sponge. That's more a dead sponge color. But yeah, that billowing stuff, I am not sure what that is. I kind of thought it's mucus that maybe just then gets gunk, dirt, sediment particles on it.
See how?
Yeah. Yeah. Yeah, it looks like we're still like right
Yeah,
on
we should
it.
be on the strike for another 80.
Yeah,
This marks the DVL reset, the US field. You know, so you can fudge it.
Roger.
Got another Spectrunculus grandis there, the giant cusk eel.
Oh, right in our
Ooh.
face. Low.
Go, buddy.
Oh, another
Up on our
giant
drill.
cusk eel.
Hello.
And exit lower screen. Thank you.
Gotta get a close up.
Wow, is that another one? Small at the bottom. Ah, maybe that second one was not another cusk eel. Erica thinks that might have been Zoarkid, the smaller one. But it is definitely a great night for Spectrunculus, the giant cusk eel. You
Yeah,
might
it's more
even
that
say it's
flocculent.
spectacularly Sorry.
Hopefully
Not
it
showing
stays.
up in the museum.
Yeah, maybe we're flattening out now.
Yep.
More flat.
Fattest.
Awesome feature.
There sure is a lot here. More antipathies. Sorry. Antipathies. Alternatipathies. Antipathies. Pink ones. And the stalked crinoids. A lot of different sea cucumbers. And just really having that 3D habitat and hard rock substrate makes for great habitat for a lot of these different critters.
Yeah, so our target waypoint is 3040, so we're almost there. So I can bring us to 030040 whenever that makes sense.
Yeah, we can go ahead and do that lateral move now.
Right. I'll wait till the end.
Take a local peek.
I know. Which means that would mean we're 150 translated.
Sorry?
That would mean we're 150 meters translated.
Looks like there's a more ridge up there, so
Yeah.
it might just be a local max.
Good.
Ben, I'm going to look at 030 here, and we can just watch the sonar real quick.
That's great.
Are you checking out where the ridge goes and what it does with the sonar?
Yeah, we're just verifying things on our map. All right, so I'm at 030 now. We can range out a little there. Oops. See 20 meters.
I'm gonna let's just add another 010 and then see.
Yeah,
Okay.
it seems, yeah, I would agree with that.
Yeah, this is probably likely.
I'll,
Thanks for doing that.
yeah, of course. Yeah, this is 010, 015 for us, so
Gotcha.
seems like that would probably take us to a steeper feature.
Yeah.
Thanks, Ben.
You will come off the slope, but
Raj. I'll just pop ahead here. Sorry, Kim. Sorry?
with some of the downtime, I was counting a lot of octopus from the octocone dive we did last week. And so when we were coming down now and you see all those white spots, I immediately started counting them just by reflex.
What number did you get up to?
I'm about 7 hours in of the 10 hours at the octocone in terms of video lengths, and I'm at 2,800 octopus.
Wow.
It just reminds me of the count on Sesame Street.
Yeah, I have a bad joke in there about hitting the mother load, you know, because they're...
And Chad, for that target A2, do you want us to keep on that depth contour and go around that little 20-meter high above us, or should we just keep along the spine as we go along?
Yeah, let's just keep along the spine.
Perfect.
I'll do that.
Another stalk sponge there on the left. That one, by being sort of amorphous like that, that might be Hyalonema biancarotum. It's one that takes a pretty rough, bumpy structure to it. But again, as with many sponges, that's a maybe diagnosis until one could collect a piece of it and find out for sure based on its skeletal structure. Ooh, that looks like another Hyalonema. I think I saw a stalk. Gosh, yeah, when we were farther, I had a stalk. So those are more tulip-shaped, live on stalks, and the stalks of Hyalonema species are bare fibers, basically, of their skeletons, which are made of glass. So that glass stalk becomes basically just a rope or other hard surface that lots of other animals like to climb up or grow on, so you'll often see anemones or zoanthids growing on the stalks of Hyalonema. Ooh, and there's another predatory tunicate. Big one, too, just next to the lasers right now. What a great mix of habitats. You've got so much diversity. Oh, hello, rat tail, grenadier. there's a lot of diversity of, mix of tall rocks, bumpy rocks, smooth rocks, sediments interspersed. A lot of different habitats in really close proximity. That grenadier is just hanging out there.
Scientist. Scientist. Can anyone hear me? Can you guys hear me?
I can hear you.
Yes.
Oh,
Some
Raj,
other
okay.
feedback.
It sounds like one of you guys has a bit of feedback on your mic. Not sure if there's a way to mitigate that or not. Or in the background, maybe.
I have been muting my microphone because of the dog barking. So I don't know if it's me, but I'm gonna mute again and then let me know if you hear me still.
Sure.
Okay, sounds like that is working for me. Okay. I think it is not my bad.
Raj.
Chad, are you there? I think it's on Chad's end.
We're getting into some smaller rubble here. You can see some salt crinoids still, anemones, a lot of different shapes of sponges. There's an interesting shape of the barrel sponge over there coming up next to the lasers now. And then also, I know I'm very sponge-centric in what I spot, but there have been also a lot of dead sponge skeletons that we've been passing over. Ooh, and now we're seeing some corals on that rock there in the distance. So I think the general consensus was that this white one is likely Narella. Oh, Tina 4 drifting by on the left side of the screen.
All right, we just crossed the 10,000 foot depth. We're a little shallower than that now as we're moving up this relatively small volcanic feature on the seafloor near Davidson Seamount. We have this dive until tomorrow late morning, I believe, and so we have a lot of ground to cover. So we're just going to be moving like this for most of the dive, but we will stop occasionally to look at things for sure.
Looks like as we come up onto the top of this ridge area, I'm starting to see some xenophyophores too. I didn't see them before, but I think it's because there's now some sediment interspersed. Oh, there's a hyalurina to the left. So xenophyophores are single-cell protists, but they're the world's largest, so they can get large enough as some of these smaller rocks here several centimeters in diameter. I'll try to point out one when it gets near the lasers, but keep an eye out for things that are a very rich dark brown and kind of look like little lumps or folds on the seafloor.
Amanda, my apologies.
Oh, there's an octopus.
Oh yeah, look at that. Can we get a quick zoom on the octopus?
Sure.
It was quiet for quite some time, so I thought no one had been speaking. So I started talking for a couple of minutes, and my microphone was still listed as not being muted. But this has happened twice in this dive so far. I had to reboot VLink, and when I log back in, I hear everyone talking. So my apologies.
All right, Marley, go ahead.
Oh, no worries. We're glad you're here. Yeah, I had some troubles with feeling too. This octopus is in a dead coral or dead sponge field. It's like
Guarding it.
Can we kill lasers for just a
Yeah.
second? This guy is beautiful.
let's see. I think
It looks like a male.
it's octopus.
Third arm.
Yeah, I would say this is
Yeah.
Bent octopus. My guess. Big eyes relative to moose octopus.
Yeah, it's
You
very
said you
closely
saw
related.
it looks like a male.
Yeah, the third arm has got the heterococylus or cotylus.
Oh, it does. There it
you
is.
go.
Yep.
Yeah, so this is a male.
All right, Marley, go and come away,
Very
please.
cool. Thank you.
Thank you. What Chad is referring to, the hectocotylus, is the third arm of a male octopus is modified to be able to deliver a sperm packet to the mantle of the female. So you can see that modified arm, then you know that it's a male. And the next question comes, where do you start counting with arms? And there is an answer to that. It's if you're the octopus, you start from the right eye, and the first arm that you see from that right eye is arm number 1, and then you count from there, 1 through 8.
Yeah, the other
Were you
genus,
able to
moose
hear us?
octopus.
When-
No, that's what happened with VLink. So there was no noise for quite some time, and then I had said a couple of statements, and then it just occurred to me, because there was no response, that I think I was having that similar error to earlier in the dive. So I just logged out of VLink and then logged back in, and immediately heard you in the middle of a story. So I realized VLink had cut off for some reason.
If it seems that you've been quiet for a long time again, I'll send you a message just to check that in the science chat to make sure. That way we can check on that. Or Hyalonema, the tulip-shaped sponge there on the stalk. I'm a fan of those. They're one of the ones that we saw really neat behaviors in.
Yeah, but Amanda, I look at those and I immediately think a very large wine glass. Does that indicate a problem?
Hey, it's all what we tend to think about. But you know, pouring wine into a sponge just sounds like a leaky proposition.
Yes.
Like.
Yeah, but it is made of glass. Sorry. Dad.
Oh, nice. I love this science humor. All those anemones pointed slightly to the left in our view, and 3 of the past stalked crinoids that I've seen were pointed the other direction. Interesting, everyone growing in the right way to get what they need from the current.
Mm-hmm.
There was a study that looked at the direction that anemones point their oral disc, the disc on the top, and they did find that that disc could be oriented into the current more often. That could be what's happening here. believe those are sea cucumbers, the pink things. I say believe just because I'm open to them being also xenocoelomorphs, xenotrabellas. I have never seen one other than photos, but I know that they can get very large and they can look like weird pink socks on the deep sea floor, and those were painted kind of floppy. But probably cucumbers. More soft crinoids all pointed one direction, and an anemone pointed the opposite direction.
Yeah, I'm finding that very interesting.
I'm thinking the suspension feeders, they might just be blown over sideways, possibly, because I see some of the corals also bowed a little bit that way. We just passed an antipathy that was that way. So for them, it might make sense to grow that way so that the water washes over them. And then for the anemones, they'd be capturing things, which means, you know, maybe they want to be facing into the currents because they're more likely to have something fly into their mouth. Oh, it's a lovely sponge. Nice sponge.
That's a cool one.
Yeah.
Very nice.
That's a relatively small Spectrunculus grandis, giant cusped eel. I say relatively because we've seen some really big ones on this dive.
Marley, gonna do a snap zoom there, please?
Sure thing.
That is the sponge you would just want to hold like a pillow, and it would betray you because they're still because they're made of glass, like holding fiberglass. Oh, wow.
There's a good current here now.
Really?
All right, Marley, go ahead and come wide, please.
Really great to get to see the structure on the outside and the inside of the osculum, that opening. You can see just how vacuous and open it is on the inside. Sponges are like a series of pipes and canals, so water passes through ever-growing and shrinking canals where it's pumped and eventually filtered to capture particles, and then it's all pumped out of that big chimney. And so the osculum is this convergence of all of those pipes, basically, emptying out into this one chimney.
A little to the west. Yeah, so Chad, I mean, we don't have to go to the waypoint, so we went up along the spine there, and then do you want us to just head to the next waypoint after this? Keep it along another ridge.
My apologies, the other waypoints were thrown together with a slightly different map when I had to modify this dive plan. So the idea right now is to kind of keep on the spine, I guess you could call it, this ridge, and then we'll have a little local maximum here, and then we'll go down a little bit. So that next null or local maximum, that's where we want to kind of head.
Octopus.
Roger. We'll just keep on the strike. So I think I'll keep us a little. It's going to start being more like 020.
Roger.
Yeah. Thanks, Chad.
Lovely bent octopus.
Roger.
It's getting even shallower than we thought.
Yeah.
I'm going to bring a 030 on the next move.
Roger.
Sorry, I haven't been talking on here too much. Just so you know what I've been up to. I'm trying to find some info about worm tubes to figure out what some potential candidates are for who makes those flared tubes like that. I was most familiar with them from, worms that are at hydrothermal vent spaces, and I'm not sure how diverse those tubes are, or kind of who else might have tubes that flare like that. So just trying to do a little bit of taxonomic investigation. Ooh, and we just
Can we
another
yeah,
yeah.
let's look at this fissure here. This is interesting.
Oh wow.
Hey, Kim, you bring your heading to 0. Oh, you got it. Never mind.
Put in 030.
Yeah, Roger. Going to bring your heading to 030, Kim, please.
Yeah, the octocone that we explored last week during our first dive was really a near perfect shaped cone. And so even though there were a lot of 3D structures, it was all fairly consistent and localized rugosity. Here, this is a much more irregular shaped basalt outflow, volcanic outflow. And so we're getting a lot of irregular shapes here. Some of these chasms and valleys and walls and things of that nature. This is pretty interesting terrain.
For the bearing? Yeah, sounds good.
Other different crunches on the left-hand side of that. Interesting terrain.
Is that for the bearing or for the step size? It looks like they only did it for the step size.
What?
Was that for the bearing or for the step size? Because it looks like it's only for the step size there. That.
I think you should have put it. Oh, that's added from the previous move. Should be 040.
Gotcha. Now there's a good current.
Right on.
Yeah, I was going to say, seems like it's slow wind. Well, doing great on time. I did some math on where we should be at each waypoint, or at least by what time we should be at each waypoint. And I said we should be at A2 by 7:15 local time Pacific.
Nice.
And we pretty much reached that, and it's about 7:11, so doing well.
So then the next move is going to also be a 040.
Roger.
We're going down slope now.
Yeah.
Great.
Yeah,
Go 040 next move there, Kim.
Once we get to the bottom, you can see the strike. We'll make it better.
Roger that. So it looks like 50 meters, 70 meters till we get to the flat and start to head up again. Roger. Yeah. The rubbly drop off. There for 0, Raj.
All right, so we're moving kind of over this localized high spot. Current is definitely a little stronger up here. But once we get over the ridge here, we'll probably drop down a little bit, maybe lose a little bit of visual, and then we'll start another ascent up a local maxima.
I'm giving up on my tube search for now. Haven't found too much about worm tubes quickly and easily. It's still excellent suspension feeder territory. You can see a lot of different animals that are made for catching things from the water column, like sponges, these stalked crinoids, corals. So right now we can see a bass patties there, just go down from the lasers. Sponges are many of the white animals. Actually, a lot of these sponges are on stalks. A lot of these are stalks that help get them up out of the water flow that's just near the seafloor. It's called the benthic boundary layer, and it tends to be slower current speeds than what you would get as you move farther up out of into the water column. So for something that feeds based on how much water passes by, getting taller can really help.
So as we're making our way through the area here, for anyone watching at home, just wanted to kind of explain how everything's been working. Usually, Ocean Exploration Trust, who runs the exploration vessel Nautilus, has a fairly long field season with partners that they work together with to plan and execute different missions around the globe. The past few years, it's really been the Eastern Pacific for the most part, although they have gone down to the South Pacific for a few missions. And typically we'll bring 6 or 7 scientists from the cooperating organizations. So the last couple of years, we've been able to take 6 or 7 scientists aboard, and we all will physically participate. But of course, because of the pandemic, they had to implement new COVID-19 mitigation strategies, which really was surprising that they could have any field season at all, considering the climate of what's going on right now. So I was able to join the expedition last week back home now, but most of the team, we've been able to direct dives from the comfort of our homes. That's why you might occasionally hear airplanes and dogs barking and cats meowing and children running by, because that's what's going to happen. So for right now, for example, Amanda and I are actually watching the stream from our houses, and we're hooked up to kind of a voice chat line that's hooked up via satellite to the Nautilus itself, and so we're able to communicate verbally with the crew that's sitting in the control van atop the Nautilus. And that includes the navigator, a couple of pilots, the video engineer, data logger, and anyone else who might be in the van. So hopefully it sounds fairly seamless, except for the odd noises you might hear back and forth. But I'm really excited for like the future. I definitely want everybody to be able to get back on a ship to conduct this kind of research, because there's a lot of stuff on the back end that I think people at home don't always see. Even though there's the camera in the wet lab, there's all sorts of sample processing that needs to take place afterwards. There's a bunch of cataloging, a bunch of other interesting things. There's just the ability to network and be on the ship and talk about things rather than just a daily meeting or 5 minutes prior to a shift. These dives also can be more than 24 hours long, which is a really great advantage of the whole system with the Nautilus and their ROVs. They're not limited to a human kind of workday of 8 hours or so. So to accomplish that, we have enough people on the ship and on shore to rotate 4-hour shifts. So, for example, Amanda and I are working the 4 to 8 shift, and we would be on again at 4 a.m. in the morning to 8 a.m. in the morning. So it happens also with a team of 2 for 8 to 12 and 12 to 4 as well. So we'll just rotate throughout the entire dive. So it does make interesting sleeping patterns, especially when we're at home and we still have to be around people that are on normal sleeping patterns. But other than that, it's been a pretty incredible way to still participate without being on the ship. And in the future, when we're all back on the ship, it can still be implemented to get experts and other people that maybe who couldn't come for whatever reason, they'll still be able to participate in this way.
It's a pretty impressive fisher